That's where we're going to spend most of our time. We will mention cardiac and smooth muscle, but this is what the vast majority of what's on this chapter and the vast majority of what's on your test, okay? Now, for muscles, there's 4 characteristics that all muscle cells have. In order for it to be a muscle cell, it has to be excitable, contractible, extensible, and elastic, okay? All muscle cells are. If a cell is excitable, contractible, extensible, but not elastic, it's not a muscle cell. If it's elastic, extensible, contractible, but not excitable, it's not a muscle cell. Hopefully you see where I'm going. Has to have all 4 of these characteristics too in order for it to be a muscle cell. What excitable is it can receive something from the outside environment. A muscle cell receives something from a nerve. It causes the muscle cell to begin to change. Contractible, it can shorten forcibly, all right, moving or pushing something. Extensible, it can be stretched, muscle cells stretch, and elastic, it comes back to its resting limb. This is a characteristic of all muscle cells. There are other cells in the body that are excitable, but they don't have all these other characteristics. Nervous system cells are excitable. We've talked about some connective tissue that's elastic, but the connective tissue that's elastic is not also excitable. So again, all 4 of these characteristics are components of a muscle cell. Now, for muscles, 4 important functions of muscles. They produce movement. They also produce some body heat. All right, they help maintain posture, and they provide stability to joints. We understand that from chapter 8. The more toned your muscles are, the more quickly they're ready to respond, the less likely joint injury. But some people don't understand, or I can't say understand, fail to realize that muscles actually generate about 40% of your body heat. All right, there's muscles. But we also know when we shiver, what happens? Muscles start rubbing and moving. Muscles start moving. It helps increase body temperature. All right, so now, skeletal muscle, all right, where we're spending most of this chapter. Skeletal muscle is an organ made up of different tissues with 3 features: nerve and blood supply, connective tissue sheaths, and attachments. Okay, we're going to talk about those, but first, let's talk about a tissue. A tissue, if you remember back in maybe early in the semester or even a general biology class, a tissue is 2 or more, organ. I'm saying tissue, I mean organ. An organ is 2 or more tissues that work together. Remember your tissues? Muscle tissue, nervous tissue, connective tissue, all right? Those are some of our tissues. So when more than one tissue works together, it becomes an organ. So muscles, this is what I want you to understand, muscles are not all entirely muscle. They're mostly muscle, but there's some connective tissue in muscle. And there's some nervous tissue in muscle, all right? And there's other types of tissues, not really a lot of epithelial tissue, but there are other tissues in muscle. So muscle are an organ. Now, each muscle receives a nerve, an artery, and a vein. Consciously controlled skeletal muscle has nerves supplying every fiber to control it, okay? Contracting muscle fibers require huge amounts of oxygen and nutrients and also need to get rid of waste products. First, consciously controlled skeletal muscle has a nerve going to each fiber. We're going to look at how small an actual fiber is of a muscle. Every muscle fiber has a nerve going to it. So nerves are permeating and stretching and interwoven all throughout every skeletal muscle. And in order for that skeletal muscle to contract, a nerve is telling that muscle what to do. And again, muscles require a lot of oxygen and energy. Now, each skeletal muscle, as well as muscle fiber, is covered in connective tissue. All right, this again, the organ. There's an epimysium, a perimysium, and an endomysium. All right, the epimysium is the connective tissue that goes around the whole muscle. Then, the perimysium, muscles are divided into smaller groups called fascicles. There's a connective tissue going around the fascicles. And then, the endomysium is the connective tissue going around one individual muscle cell. Let's go to a picture, we'll come back. Here's a picture. This is a good picture, you guys, okay? All right, so here's a picture, all right, which shows us epimysium, all right, perimysium, and endomysium. First, this muscle here, the tissue around the outside of it that holds all these muscle cells together is the epimysium, connective tissue. It's not muscle tissue, it's connective tissue, like glue that holds together. The dense, thick tissue. Then, each one of these little, it looks like it's divided into little segments. Each segment has a tissue going around it. This is one segment sticking out here. So around this whole segment here is a perimycete. And this segment, which is a group of muscle cells, this is one individual muscle cell. Muscle cell is also called a muscle fiber sticking out. And around the muscle fiber is what's called an endomycete. So our muscle is arranged in these unique groups. Backing up. Epimysium covers the whole muscle. Then the muscle is divided into little kind of groups called fascicles. All right. For instance, if you flex my big bulging bicep, it feels like there's little strings and strands in there. You know, when I flex, I feel like a whole. All right. All right. The little strings and strands in your muscle, the divisions, those are often fascicles. So, whole muscle, epimysium. Individual groups of muscles, perimysium. Each individual muscle cell, endomycete. This is the connective tissue surrounding it. All right. Back to the picture there. Is that making sense? All right. Now, as I mentioned, this is a good picture. When you looked at muscles in the beginning of the semester under the microscope, you won't pull out a microscope again, but this is what you were looking at right here. So this edge right here where my finger is, that's the epimysium. Notice it's divided into groups here. Those are fascicles. This space around the fascicles is the perimysium. Zooming in each fascicle is multiple muscle fibers, which would have endomysium around those. Okay? Muscles, all right, they span joints or they go across joints to permit movement. All right, muscles attached to a bone in at least two places. There's an insertion and there's origin. Insertion is the attachment to the movable bone. Origin is attachment to the non-movable or less movable bone. We talked about that already, right? In chapter 8, and I flexed my bicep for you guys. All right, so you guys don't need me to flex it again, because if I did, you might pass out. So, all right, you got that. Attachments can be direct attachment, fleshy. The epimysium is fused to the periosteum of the bone. Or indirect, you have a connective tissue. Sometimes there's a rope-like tendon or a sheet of connective tissue that's indirect. Sometimes the muscle is directly attached to the bone. You don't spend a whole lot of time on it. Back to the picture here. And you can see here this is a tendon, which would be an indirect attachment there. All right. On the bus, happy, smiling, joyous. All right, big plans for the weekend. I might be free. Just send me a message. I'll pull up. I might give you some gas money. Right? If you let me ride with you. Anyway, never mind. All right. So muscles. All right. This is showing the whole muscle. This is showing a fascicle. This is showing a muscle cell. All right. A whole muscle, again, wrapped around there is the epimysium. The one unit or group of muscle cells sticking out, this is called a fascicle. This shows us the fascicle. Inside a fascicle, there's bunches of little muscle fibers, muscle cells. Remember, a muscle cell and a muscle fiber are the same thing. The book and people who study muscles use those terms interchangeably, okay? No difference between a muscle cell and a muscle fiber. But here's an individual muscle fiber or cell. In the individual muscle fiber or cell, you see this one little rod sticking out. All right, that's called a myofibril. It's not a cell, it's just a part of a cell. And inside each individual muscle cell, there can be hundreds to thousands of myofibrils in an individual muscle cell, okay? The more myofibrils in the muscle cell, the stronger the muscle cell. So when we, like me, go to the gym to build muscle, usually around here, all right? When we go to the gym to build muscle, all right? You usually have, most people have a genetic limit in terms of the amount of muscle cells they can have. It varies. Now, anywhere, how things have a range between, somewhere between 80 and 120, or it's a range, right? But as you increase muscle strength, what you really can do is you can increase the number of myofibrils in a muscle cell, but you don't literally increase the number of muscle cells. Let me back up. You can increase the number of muscle cells slightly. So let's say, for instance, I've never been to the gym. I got 80 muscle cells in my bicep. I start going to the gym, I might get to 120. But 120 is pretty much genetically about all I'm going to get. But somebody else, like let's pick on Tristan. Tristan might have 120 muscle cells, and he might be able to go to 150. Genetically, there's slight differences in the number of muscle cells an individual can have. That's related to genetics. But what we do in terms of our working out is we increase the number of myofibrils in our muscle cell. And the more myofibrils in the muscle cell, the stronger the muscle. For instance, there are some people who don't have the mass of a hulking, huge bodybuilder, but they're stronger than a bodybuilder. And that all has to do with the number of myofibrils in the actual muscle, not the size of the actual muscle. Does that make sense? Mm-hmm. So anyway, we're going to spend a lot of time talking about myofibrils and how myofibrils play a role in creating a contraction. Okay. All right. So skeletal muscle. All right. The fibers are long cylindrical cells that contain multiple nuclei. Skeletal muscle fibers, they can be as long as a foot or so in length. One cell has multiple nuclei, but can be a long cell. All right. And when they contract, that 12-inch cell can shorten to 6 inches, and that provides that forcible contraction. Now, some words to understand and know, all right? Sarcolemma, sarcoplasm, whenever you hear sarco, that relates to muscle. Sarcolemma is the muscle cell or muscle fiber plasma membrane. If you think about the plasma membrane of a cell, remember all cells have boundaries around them. Muscle cell has a boundary around it. We just don't call it a plasma membrane, we call it a sarcolemma. Long story behind that, which relates to botany and botanists, and you guys don't want to hear about a botany story. It's just called the sarcolemma. Okay, that's the muscle cell membrane. You won't hear anyone say muscle cell membrane. You won't see on the test muscle cell membrane. You have to be able to relate muscle cell membrane and sarcolemma are the same thing. Okay, sarcoplasm, that's the cytoplasm. Just like there's a cytoplasm, there's a sarcoplasm. So we have unique names for the cytoplasm in muscle. Now, muscle has a lot of glycosomes and glycogen storage and myoglobin for oxygen storage and some modified organelles, myofibrils, sarcoplasmic reticulum, and T-tubules. And we'll spend time talking about that. We mentioned myofibrils already. We'll see them again. And sarcoplasmic reticulum and T-tubules also play a role with the myofibrils. All right. On the bus. This is the chapter everybody loves, really most people hate. All right. It's a lot of weird terminology that you probably never come in contact with. All right. And you have to kind of understand the terminology in order to understand what happens in muscle cells. And for some reason, the state of Alabama wants me to teach you all these wonderful details inside the muscle cell and what happens. All right. Questions on the bus? Happy, smiling, joyous, good. All right. Myofibrils now, inside the muscle cell. They are densely packed, tightly connected together, rod-like elements. A single muscle fiber can have thousands, accounts for 80% of the muscle cell volume, so the vast majority. Myofibrils have striations. Remember how muscles have the lines? The myofibrils are responsible for that. They have sarcomeres that we're going to talk about. Sarcomere is the smallest unit of a muscle that contracts. Myofilaments, we'll talk about actin and myosin. These are proteins. And the molecular composition of the myofilaments we'll touch on briefly. All right, this shows again a big blown up picture, large picture of an individual muscle cell. You can see the striations here. You can see kind of peeled back right there. That is the sarcolemma. We can see these small rod-like elements on the inside. Those are called myofibrils. And if you look real close, you can see the myofibril has all these different divisions or connections in the myofibril. I wish I had more. I should have brought my sharpies. Anyway. We should have more than 2 sharpies in the classroom. Those on 150 times. Let's just pretend all these sharpies connect together. They don't. All right. But all these sharpies, let's pretend are myofibrils. Okay. So you have a myofilament, all right, as we're looking at here, and the myofibril has all these different units in it. What happens when the muscle contracts is that these fibrils all shorten. So this, let's say this is 12 inches, will then shorten to just 6 inches because it's shortening. This is how, when you're lifting weights or moving something, this is how muscles are pulling something. And we're going to talk about how they shorten. And then when you're relaxed, what happens is they come back to normal. So they shorten, contracted muscle, they go back to normal size. And what is happening is these individual myofibrils, and there's small proteins in there. And so imagine this. All right. So there's small proteins in here, but let's just say each one of these is a protein. So let's say that's 10 proteins there. But then I got 1,000 of these. So 10 times 10,000 is 10,000 proteins. So when I get ready to lift something, I have 10,000 proteins in one individual muscle. Really, it's more than that. All pulling on the same force. So back to what I was saying. When I increase the number of myofibrils, instead of 10,000 proteins, now I got 11,000 proteins. So now I'm stronger. Okay? All right, that's kind of where we're going with this. These are okay. So now the striations, you guys, they have names. There's A bands and I bands, all right? Dark bands and light bands. A bands are dark bands. So you think about what's the vowel in the word dark? It's an A. So whenever you hear of a dark band, it's an A band. Or whenever you hear A band, it's dark. I bands are light bands. All right, what's the vowel in light? I. All right, so that's kind of one way to kind of remember which one's which. If you're asked the question about an A band or an I band, which one's dark, which one's light. Now, dark bands are dark regions, light bands or I bands are lighter regions. There's also what's called an H zone and an M line and a Z disc that we'll see in just a minute. The Z disc, from one part of the Z disc to the other part, the distance between two Z discs is one sarcomere. We'll see that in just a minute, okay? So this shows your A bands and I bands together. And if you look real close, between these bands, specifically these light bands, there's a thin other line in there also. And you see, let me skip here. You see this here? For instance, dark band, A band, where the red is, and specifically the red and the blue are overlapping, those are proteins. So where you have overlapping proteins, it's darker in color. Where you have only one protein here, that's the light band, it's lighter in color. So more material proteins, it's darker. Less material, it's lighter. That's what forms the striations that we see in an individual muscle cell. So here's your I band, there's your A band, all right, again. And basically, this zigzag line is a Z disc. This blue zigzag line over here, so between these two blue zigzag lines, that is a Z disc, two Z discs, and the distance between two Z discs is one sarcomere. Okay? So Z disc, one sarcomere, and what makes up the sarcomere, there's the little blue and the red proteins, and the blue and the red proteins are responsible for helping the contraction that occurs. We'll back up because I skipped something. We did that, we skipped this. Okay, a sarcomere, I mentioned it to you in the picture already. It's the smallest contractile unit. It's the functional unit of a muscle cell. Contains an A band with half of an I band on each end. Also, it's the area between two Z discs, that's a sarcomere. Individual sarcomeres are aligned end to end, like myofibrils, or like on a myofibril, like boxcars on a train, and back to the picture here. Okay. So, a sarcomere, to show you where we're going and what's important with sarcomeres. All right, there's the blue protein, which we're going to talk about, is called actin, and there's the red protein that you see there, which is called myosin. These proteins overlap just a little bit when the muscle is relaxed. When a muscle contracts, the proteins do not shorten, but they overlap. They slide over each other. It's called the sliding filament model of contraction. So let's pretend the green is my blue, the black is the red. What happens is it does this. And so the proteins, as you're contracting the muscle, they slide back and forth to cause the sarcomere to shorten. The proteins themselves are not shortening, they're just overlapping one another. We're going to look at that in a minute, okay? All right, so the myofilaments, orderly arrangement of actin and myosin myofilaments within the sarcomere. Actin myofilaments are called thin filaments. The myosin myofilaments are called thick filaments. If you back up to the picture, actin was the blue, myosin was the red. They extend across the I band, partway to the A band. They're anchored to Z disc. The actin is. The myosin extend the length of the A band. They're connected at the M line. I'm gonna back up to that picture again. So when you're looking here, red is the myosin. It's connected right here in the middle. The blue is the actin. It's connected to this zigzag line. And so when the blue gets pulled over the red, the zigzag lines, the Z discs get closer together. That is what the basics of a contraction. All right, when you look here, this is a more realistic picture. The thick filament is the red, the thin filament is the blue. The red has all these little knob-like, considered hands. They got a bunch of hands that reach up and grab onto the blue. So when you contract a muscle, all these hands start pulling on the blue. And when they pull on the blue, they pull the blue closer to the M line, closer to the middle to create a contraction. This here, Z disc, that's a Z disc. This is one sarcomere. Okay? Everybody still with me? Happy, joyous, smiling, good. All right, now, what you're looking at here is showing you your myosin, the thick filament. You see 1, 2, 3 thick filaments. You see the blue filaments. This gives it in a wonderful 2D view, which is easy for us to kind of think about. But in reality, this is in 3D. So when we look at it in reality, if we just skip here, there's a red surrounded by 6 blue. There's another red surrounded by 6 blue. So when we're contracting, what's happening is it's pulling here and pulling here and pulling here and pulling here. It's pulling on 6 different fibers, all right, at one time. It's what's really happening. So you don't have one red filament, myosin, for one blue filament, or 2, if you're looking here, because it looks like it is a 2-to-1 ratio. It's not really a 2-to-1 ratio. It's really a 6-to-1 ratio. And that's why you have all these little myosin heads that they call, and they're pulling in multiple different directions at any given time of a contraction state. All right. So now, with these myofilaments, all right, and again, when you say myofilaments, you're talking about the actin, the thick filament, the actin, which is the thin filament, and the myosin, which is the thin filament. Thick filaments are made of a protein myosin, all right, and basically this other stuff we're not going to talk about. We're not going to talk about 2 heavy and 4 light polypeptide chains, myosin tails, and globular heads. We're not going to talk about any of that. We will come back and talk about cross bridges. But a cross bridge is when the thick filament and the thin filament connect. When they touch, that's called a cross bridge, okay? Other than that, we won't talk about any of that. And this is our thin filaments, and we're not going to talk about the globular units and F-actin, G-actin, and all that stuff. We're not going to talk about it, all right? Again, the cross bridge is when these 2 filaments connect. We will talk about tropomyosin and troponin, though, okay? They are regulatory units. When I say regulatory, they kind of control when actin is active and when actin is not active. All right, I messed up the goal. So here's a picture of the thick filament with all these myosin heads. Here's a picture of the thin filament. It's made of individual units. What I do want you to understand, won't really be on the test, but to understand why this works, why the thick and thin filaments work the way they do, is the thin filament here, you see how they have these little indentations here? Those are like handles for the myosin heads to grab on. So when they start sliding over each other, the myosin heads are grabbing the handles on the blue, and it's pulling kind of a tug of war, like a rope. All right, those are the handles it grabs onto. As we talk about, this will come up later, and that's going to be part of your short answer question, but I'll explain it again. The troponin and tropomyosin. Tropomyosin is the long string, gold-like string. Troponin is the little yellow structures. Troponin and tropomyosin, they block these handles. So when your muscle is relaxed, the handles are blocked and there's no contraction. When you choose to contract the muscle, what happens is troponin and tropomyosin move out of the way, then the red grabs and pulls and creates a contraction. Then when you relax, you're blocking the thin filament. Contract, you opened it up to be grabbed onto. And this is one of the things we'll see, and we'll see this in the video. All right, on the bus. All right, okay. There's some other stuff that I'm gonna cover these next slides real quick because this is really a test. What we're gonna explain to you, we're talking about actin, we're talking about myosin, we're gonna talk about ATP, we talk about the sliding filament. But muscle contraction and what muscles do is way more complicated than that. There's other proteins like titin and dystrophin, nebulin, myomestin, and C proteins. We're not even gonna talk about those. I want you to know there's other proteins, that's it. Those won't pop up in terms of understanding what they do. A disease associated with that, we don't talk about dystrophin, but muscular dystrophy is a disease often seen in boys at a young age. And again, the gene has to do with dystrophin. We'll skip that. This is a young man doing some physical therapy who has muscular dystrophy. And these are some of the symptoms of muscular dystrophy. The good thing is that patients are now living much longer with muscular dystrophy than they used to. All right. The sarcoplasmic reticulum. Everybody heard of endoplasmic reticulum before? Remember that? Sarcoplasmic reticulum is the endoplasmic reticulum of a cell. All right. But it's the endoplasmic reticulum of a muscle cell. All right. Most run longitudinally. The terminal cisterns are a part of the sarcoplasmic reticulum on the edge. All right. And the sarcoplasmic reticulum, that's what SR stands for, functions in the regulation of intracellular calcium levels, and it stores and releases calcium. The main job of the sarcoplasmic reticulum in muscle cells is to store calcium and release it when it needs it. And then it stores it again, and it releases it when needs it. And it stores it again, and releases it when needs it. Sometimes what happens in our bodies when you have a muscle twitch, all right, or even a cramp, sometimes you damage the sarcoplasmic reticulum, not the whole cell, not the whole muscle cell. Then now calcium is no longer being regulated, and you have to wait for the heal itself. Anyway, well, it'll make more sense in a minute. T-tubules are another structure. A tube formed by protrusion of the sarcolemma deep into the cell interior increases the muscle fiber surface area greatly. The lumen is continuous with the extracellular space, allows electrical nerve transmissions to reach deep into the interior of each muscle. We're going to explain this in just a minute. Main job of the T-tubule is that it allows for the signal to contract to go deep inside the muscle. We'll explain that. We'll see a picture to help explain that. T-tubules penetrate the cell's interior at each A-I-band junction. Remember we saw A-bands and I-bands? So wherever there's an A and an I-band in a relaxed muscle, there's a T-tubule right there also, okay? And where the T-tubule is, there's something also called a triad, which is the terminal cistern, and a T-tubule, and another terminal cistern, okay? So, let's see. I'm gonna skip to the picture to show it. This is showing you a muscle. I wanna show you the triad, okay? I skipped ahead a few pictures, I'm gonna back up. This kind of gray or white tube, this is the T-tubule, okay? On the edge of the T-tubule, these little blue tubes, that's the terminal cistern, okay? The terminal cisterns connect to all of this, which is the sarcoplasmic reticulum. So the triad is terminal cistern, T-tubule, terminal cistern, okay? These are basically what you're looking at, all right? And basically what this also forms is the T-tubule, when you're looking at the A-I-band junction, it's about the distance of a sarcomere, okay? Approximately, not exactly. So that's what you're looking at. So we move back up to here. The triad is a terminal cistern, which is just your sarcoplasmic reticulum, and a T-tubule, and another terminal cistern, or another sarcoplasmic reticulum. So it's when the sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum come together, that forms what we call a triad. triad relationships. So in terms of what it does, the T-tubule contains these special proteins that stick into the intermembrane space. And these proteins, when they see that the muscle, when they see that the nerve tells the muscle to contract, these proteins help release calcium, okay? And calcium is important for muscle contraction. And again, we will see that in a minute. When an electrical impulse passes a T-tubule, proteins change shape, causing the sarcoplasmic reticulum proteins to change shape, causing the release of calcium into the cytoplasm. To the picture here. Now, let's kind of explain using this picture what's taking place. First, we know that the two filaments slide over each other, right? And do you remember there is troponin and tropomyosin blocking the thin filament? Remember that? Okay. Calcium causes troponin and tropomyosin to move out of the way so that they can pull on one another. Whenever there is no calcium or low levels of calcium, there's no connection between the two fibers. So calcium is the key that causes the connection. You with me there? All right. So calcium is the key that causes the connection. Now let's bring T-tubules and sarcoplasmic reticulum in. We're going to talk about it as we keep going today. The brain tells the muscle to contract. Skeletal muscles are under your voluntary control. So when your brain tells your skeletal muscle to contract, it sends a signal that goes to this membrane around here called the sarcolemma. You can kind of see this little membrane here. So when the brain sends a signal to the sarcolemma, the signal starts moving around the sarcolemma. As it moves around the sarcolemma, it comes to a T-tubule. It goes up or down, doesn't matter. The T-tubule. When it goes up or down the T-tubule, the terminal cisterns, the triads, see that. When they see the signal from the brain in the T-tubule, the terminal cisterns cause the sarcoplasmic reticulum to release calcium. You with me? Now, if you look here, the red and blue, remember the red and blue filaments? So this blue has troponin blocking it. When the sarcoplasmic reticulum releases calcium, the troponin are moved. So now the actin and myosin can contract. This is how our brain sends a signal to the muscle fibers, which then go to the myofilaments to cause them to contract. Isn't that fun? You guys really don't want to hear about anything. I know, I'm sorry. But that's a good portion of the chapter. And this also is a portion of what your short answer question is. I'm going to go over it kind of as much as I can before the end of the day today, and let you record me do that. But that's the type of stuff you're going to have to do. And for instance, to give you kind of an example, on your short answer question, I will give you the words, so you get a word bank. I will say, please explain skeletal muscle contraction using the following words: sarcoplasmic reticulum, T-tubule, actin, myosin, calcium. I'll give you a bunch of words, and you have to explain it and write it out, and tell me what's happening. 20 points. And remember, I will repeat it for you today, let you record it. I'll do it next class, and as many times as you want me to up until the day of the test. I'll record it. You can pull it out, your cell phone, record me, and I'll repeat it over and over and over again. And if you verbatim give me back what I tell you on the test, that's 20 short answer points. Okay? But if you understand the T-tubule, and understand the calcium, and understand all those good processes, then you guys, you're also going to get some of the multiple choice questions right on the test. So it's good to, I would safely say that if you know the short answer question and know what each of those things do, those words, what they do and how they function, I'd safely say you got 50 already. But that, I haven't asked any other questions. 20 points, there's probably another 30 points in the test, just by understanding that. The other stuff we teach in this chapter on muscles is kind of simple, in my opinion, much more straightforward. Okay. On the bus. All right. All right, so contraction is the activation of the cross bridges to perform and generate force. Only time a cross bridge occurs is when actin and myosin can connect. Remember, we mentioned that. We'll see cross bridge later. But whenever actin and myosin connect, the red and the blue filament, when they touch, that's a cross bridge. Shortening occurs when the tension generated by the cross bridges on the thin filaments exceeds the forces opposing shortening. Contraction ends when cross bridges become inactive. So when we inactivate and relax our muscle, what happens is we remove the calcium from the muscle, and the troponin and tropomyosin are now blocking the thin filament. In the relaxed state, the thick and thin filaments overlap slightly at the ends of the A band. The sliding filament model of contraction, this is what we're explaining to you, all right, states that during contraction, thin filaments slide past thick filaments. I showed that to you with the markers already. Neither thick or thin filaments change in length, they just overlap more. When nervous system stimulates the muscle fiber, the myosin heads are allowed to bind actin, which I just explained. That forms a cross bridge because now these two filaments are connected, and that causes the sliding process to begin. All right, when this happens, all right, Z discs are pulled toward the M line, I bands shorten, Z discs become closer, H zones disappear, and A bands move closer to each other. I won't really talk about that, but you can see it here. This is a relaxed muscle. Notice Z disc, thick, thin filaments, light band, dark band, light band, all right? Here is a contracted muscle fiber. Now, notice the Z disc here, contracted. Back up, notice the Z disc there. So again, imagine thousands of these little units, sarcomeres. When thousands of these sarcomeres contract, you have tens of thousands of proteins, all right? In 1,000 myofibrils, which is again creating force for the shortening. So again, relaxed, oops, wrong way. Relaxed, contracted, relaxed, contracted, relaxed, contracted. So this is what's happening. But it's happening hundreds of thousands of millions of times. All right, and this is what gives force to the skeletal muscle contraction. On the bus there. Yes, ma'am.
Do you say that would be important for us to be able to tell the difference between those muscles for the test?
Between
Between the relaxed and contracted and all that?
no, I don't think there's anything on the test there. I would want you to know that the relaxed is a slightly overlapped, and the contracted has a greater overlap. And deeper than that,
Just like the, write out difference between them and not the picture.
yeah.
Thank you.
You're welcome. All right. Now, muscle fiber contraction, you guys. Decision to move is activated by the brain. Signals transmitted down the spinal cord to motor neurons. Neurons and muscle cells are excitable cells. We already mentioned that, capable of active potentials. Excitable cells are capable of changing the resting potential, and the active potential crosses from the neuron muscle cell by the neurotransmitter acetylcholine. So we're going to add to what we've been talking about. Right now, what we were talking about is what happens inside the muscle cell. Now we're going to talk about what happens outside the muscle cell for that to happen inside the muscle cell, if that makes sense. So neurons, all right. Motor neurons, they have axons. Each axon has a lot of branches. They connect at a place called the NMJ, Neuromuscular Junction, all right? Another name for neuromuscular junction is motor end plate. And each muscle fiber has one neuromuscular junction with one motor neuron. We see that here. This young lady's scrolling on her phone, all right? And as she's scrolling, she's telling her muscles what to do. And you see here's the nerve that comes to her muscle fibers. And this nerve connects with 3 muscle fibers. Now in reality, every muscle fiber of our body would have a nerve connecting to it. But one muscle can have more than one nerve, all right? But each muscle cell has a nerve connecting to it, or an end of the nerve connecting to it. What we're going to talk about is what the nerve does to cause the muscle cell to contract. So, when this young lady sends a signal down her nerve to cause this muscle to contract, these 3 fibers contract. The other fibers do not contract. When you contract your muscle, every fiber does not contract all at the same time. Usually it doesn't. Now, if you're doing a very strenuous physical exertion that requires every fiber, yeah, you will. But for scrolling on your phone, no. All right, axon terminal, you guys, this is a part of the nerve. All right, axon terminal is where the nerve and the muscle fiber kind of come together. It's a gel-filled space called the synaptic cleft. It's stored with axon terminals, have these synaptic vesicles that have something called acetylcholine. The muscle cell, the sarcolemma, has junctional folds, okay? The axon terminal, all right, it has what's called basically an axon terminal, or also called a terminal bouton, all right? The junctional folds have ACH, acetylcholine receptors. All right? The axon terminal releases a neurotransmitter called acetylcholine. And the NMJ, neuromuscular junction, is made of axon terminals, synaptic cleft, and junctional folds. Let's try to make this make sense with the picture here. So here's the picture. The gold structure you see at the top, kind of shaped like a, I don't know, what's that, a bell shape? What would you call it? Bell shape? Everyone know? Not a bell? I don't know. Whatever you call that. The gold thing, all right? That's the end of the neuron. Down here, where you have all these wrinkles, those are the junctional folds. This is the muscle cell. So, neuron, muscle cell, the space between is called the synaptic cleft. So the nerve and the muscle do not physically touch. It's a microscopically small space between the two called the synaptic cleft. On the nerve, or in the nerve, we have these little yellow balls. That's the chemical acetylcholine that tells the muscle to contract. On the junctional folds, you have these purple proteins. That catches the chemical, or the receptor for the chemical. All right. So when we tell our muscle to contract, the nerve sends a signal all the way down to the end of the nerve, which is called the axon terminal. The axon terminal then releases acetylcholine. Acetylcholine floats across the synaptic cleft, attaches to these receptors, opens the receptors, and then the signal from the nerve now is transmitted to the muscle, and this signal now goes to the T-tubule. And the T-tubule causes the release of calcium. And calcium connects to troponin. And then actin and myosin can cause the sliding filament model of contraction to occur. On the bus. You guys are just loving this. I'm enjoying this so much. Seriously, questions? All right, the big picture, you guys. 4 steps must occur for skeletal muscle to contract. There's events at the neuromuscular junction, all right, which we were just talking about. Muscle fiber excitation, we're going to talk about. Excitation, contraction, coupling, we also got to talk about that. Cross bridge cycling, we've already talked about. Cross bridge, actin, myosin connect, they slide. Events at the neuromuscular junction, we're going to talk about some more, but that's the release of acetylcholine that attaches to the NMJ, neuromuscular junction, or junctional folds, causes that signal to be transmitted. So this is what we're talking about. All right. Now, if you're looking for it, I know some of you guys are saying, just give me all these steps, just so I can go on this study, right? This here, figure 9-2, or sorry, 9-7. This summarizes those steps. To me, I think it's confusing for you guys, the figures. But if you're looking for one place in the book that kind of summarizes almost everything for the short answer question, this is it right here.
Almost everything?
Almost. But again, figure 9-7, what this is explaining, gets to the end of the nerve. When you get to the end of the nerve, the acetylcholine is released, calcium comes in to the end of the neuron. We're going to explain that in just a minute. Calcium causes the release of acetylcholine. Acetylcholine attaches to the receptors in the junctional folds. The receptors in the junctional folds open. Sodium comes into the muscle cell. Sodium causes a change in the action potential. The action potential from the neuromuscular junction spreads around the sarcolemma to the T-tubules. That same action potential goes down the T-tubule, comes in contact with the terminal cisternae. That action potential then causes the release of calcium. The release of calcium from the sarcoplasmic reticulum attaches to troponin, moves tropomyosin, and then the myosin heavy chain can attach to the actin light chain and cause the shortening of the sarcomere. That's what this is explaining, and that's essentially, in a very simple form, be sure that's your question. All right.
That's the simple form?
Yes.
Oh, Lord.
All right. Questions. All right. So this is explaining again part of the process. So this is just what's happening at the neuromuscular junction, giving you 6 steps. The action potential arrives at the axon terminal. Your brain sent it down the end of the nerve. What happens is calcium channels open at the end of the neuron. This is in the nerve. Calcium comes in. Calcium causes acetylcholine to be released. Acetylcholine floats across, they use the word diffuses. It floats across the synapse. It binds to the receptors, allows sodium to come in. Acetylcholinesterase degrades or removes acetylcholine. I don't really focus on that last step, step 6. But this is what stops, you do need to know this for the lecture exam, but not for the short answer question. This is what stops skeletal muscle contraction. Because think about it. If you're releasing a chemical to cause the muscle to contract, what stops the muscle from contracting? You have acetylcholinesterase, which is destroying acetylcholine. Acetylcholinesterase should be a spelling word, right? Yeah.
What is it?
Acetylcholinesterase should be a spelling word, right?
Give us extra credit if we can spell that.
Don't put it on
All right.
Next.
No, put it on the test. Give us extra credit for spelling.
It's not a spelling word, but so this is showing you calcium coming in, causing acetylcholine to be released. Acetylcholine is going to attach to the receptors in the junctional folds, and that's what it's showing you there. When acetylcholine attaches to the receptors in the junctional fold, the little purple protein, which is called a ligand, Ligand, a ligand-gated receptor. Ligand is chemical. The chemical gated receptor opens. When that opens, sodium goes in, a little bit of potassium comes out, but there's like 10 times more sodium goes in. And since way more sodium goes in, that begins to change the action potential. And then acetylcholinesterase comes and chews up the acetylcholine to stop the muscle contraction. Okay? Ready for a video? Which is going to summarize the process that we just went over. Questions? All right. No.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Questions? All right. So, with that, let me just back up. Just stay here for a second. With the muscle cell membrane. All right. So let's pretend this black line. These two black lines. This is our sarcolemma. All right. Inside the muscle cell, it's negative. Outside the muscle cell, I just put plus 90. I don't really know what it is outside, but it's positive, okay? Negative and positive have to do with the ions that are there. Anybody remember ions from like high school biology? Right? A little bit about ions. We're not getting into the deep chemistry of this, all right? But a positive ion is positive. Negative ions like chlorine and hypophosphate and chlorine, those are negative, all right? So you got negative ions inside, don't worry about what they are. You got positive ions on the outside. You have significantly more positive ions on the outside. A lot of that sodium. So, all that salt you put in your food, all right, it's floating around your cells, okay? So, when this process happens, acetylcholine's released, it goes to the ligand-gated receptor. What happens is in the membrane, there's an opening or a hole, okay? When the hole opens up, the sodium goes in because of diffusion. Anybody remember diffusion? Diffusion is the movement of chemicals or particles or molecules or something from a high concentration to a low concentration. So because this is a high concentration of sodium and this is a low concentration, whenever these little things open up, because acetylcholine was released, sodium goes into the muscle cell. Lost anybody there? So if sodium goes into the muscle cell, sodium has a positive. What happens inside the muscle cell is now minus 90 becomes minus 80. And minus 80 becomes minus 70. And does that make sense? So as sodium keeps coming in, this keeps going up, 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 up. It really maxes out at plus 30. This is what's called an action potential. So this is why a muscle cell is an excitable cell. Now, with this happening here, remember, right here, all we're talking about is the neuromuscular junction, or what is called the motor endplate. So, when this happens here, I'm gonna put N and J. This happened in the neuromuscular junction. What happens is it starts spreading, and I'm teaching slides ahead now. So hopefully we'll be able to cover some slides faster. And it spreads. And it spreads until it gets to a T-tubule. So let's say this is a T-tubule. So it spreads so it gets to a T-tubule. The T-tubule is not the membrane, but the T-tubule goes down inside the cell. So when this signal goes down the T-tubule inside the cell, then calcium is released, which attaches to troponin, moves tropomyosin, and causes the shortening of the sarcomere. Okay? Yes.
The plus 30, is that what happens? So the outside drops to plus 30 or the inside?
The inside
raises?
drops to plus 30.
Okay.
And I put that in the wrong place, but this goes
It'll go all
80,
the way.
70, all the way. In
All the
fact,
way
let me-
plus
let me
30,
just
okay.
show you. This is where we're going.
Gotcha.
This is an actual muscle cell contraction when you measure it.
Okay.
If you're using electrical instruments and to measure muscle cell contraction, here's what happens. You start at minus 90, and then it goes up to plus 30, and then it returns back to minus 90. Now, if the inside goes from minus 90 to plus 30, outside is really more than plus 30. I don't know what it is, okay? I'm just honest, I don't know. I can go figure it out, but I don't care. And it probably varies based on each person. But it goes 2 plus 30, and this creates a contraction. If all of this doesn't happen, there's no muscle contraction. And then to show this, what happens, you guys, look at how quick this happens. This is in less than 15 milliseconds, thousandths of a second. So in 15 thousandths of a second, the muscle contraction has occurred. Okay, so this is happening super duper fast, faster than we can really imagine. But it goes from minus 90, which is resting, to 30, contraction. And in this process, since we're here teaching, kind of help us move ahead a little bit. Minus 90, when you get to this point, which is like a plus, not my plus, but a minus 70 or so, this is what's called threshold. Threshold means this. Anything, sodium that comes in and moves from 90 to 80 doesn't create a muscle contraction. When sodium comes in and reaches minus 70, then you automatically gonna get a muscle contraction because all of the sodium channels gonna open. Right here, you got a few, then boom, all of them open. Sodium rushes in, which is why you get this sharp increase. Then the sodium channels close, and it's returning back to normal. And once it returns back to normal, then you can repeat the whole cycle of another muscle contraction. All right, let's do some practical stuff. All right, why does this make sense? All right, let's talk about that. Because you guys are like, I hate this stuff.
It don't make sense.
It don't make sense. Okay, well, why is it important? Let's talk about that. This is important because many toxins, drugs, diseases interfere with intervention of the neuromuscular junction. Myasthenia gravis, ever heard of that? The disease? All right. It's characterized by drooping eyelids, difficulty swallowing and talking, and generalized muscle weakness. What happens, there's a shortage of acetylcholine receptors in this disease. They believe the autoimmune system, your white blood cells and antibodies and stuff, attack these receptors. And when it destroys the receptors, your brain is sending the signal saying muscle contract, muscle contract, muscle contract. But there's no acetylcholine receptors. So even though you're releasing acetylcholine, the muscle cell can't receive the acetylcholine, therefore you don't get a contraction when you tell yourself to contract. So there is a practical importance to this, and we're not just studying this stuff because I want to stand here and talk about this stuff. Okay? And so there's medicines and diseases related to this. Trying to think real quick. Yeah, there is, even for instance, anybody heard of a muscle relaxer?
Yes.
There's a certain, thinking of something specific, can't come up with exactly, but there's muscle relaxers that slow this process down. So now, you know what, even though you're wanting to contract your muscles, you're like, you're just kind of droopy. And this is what muscle relaxers are doing. All right, there's some, they do different. Muscle relaxers do all types of different things. They have different ways to make them work, but this is one of the ways. All right, so this is not just some senseless mumbo jumbo that is completely unrelated to life. This is related to life, and many of you guys probably taken a muscle relaxer at one point, or will take a muscle relaxer at one point, and you could say, hey, I know what's happening. All right, on the buttons. I just explained this part to you guys, so hopefully we can go over this quick. The resting sarcolemma is polarized, negative 90 on the inside, positive on the outside, all right? Action potential causes changes. There's 3 steps. The end plate potential, which we saw, acetylcholine attaching to the ligand receptor. There's depolarization and repolarization, which we saw in the diagram there. This explains all the detail of this. I'm not gonna talk so much about this. This is part of your short answer question now. Acetylcholine is released, binds to the receptors, chemically gated sodium comes in, some potassium goes out. Because sodium comes in, the interior becomes less negative. That's what I drew here, okay? This shows the picture of that. Acetylcholine goes here, starts spreading. Depolarization is going from -90 upward, more towards zero, becoming more positive. If in-plate potential causes enough membrane voltage, reach critical level called threshold, all right, which I mentioned, -90 to -70 threshold. When that happens, a large influx of sodium comes in. So at first, little bits of sodium are coming in. When you reach threshold, it is a massive amount of sodium that comes in. The action potential spreads across the sarcolemma, so it leaves the neuromuscular junction, starts going around the sarcolemma, goes down the T-tubule. Here, as you see it spreading over here. And then repolarization is when you're returning from +30 back to -90. When that happens, sodium voltage gated channels close, potassium efflux, potassium begins to come out. There's something called a refractory period, where the muscle fiber is no longer stimulated or cannot be stimulated until it returns to normal. And there, the sodium channels close, and brings us back to the diagram we were looking at here. So, resting potential, depolarization, which is a word you should know, is going away from resting potential or becoming more positive. Or sometimes they use the word less negative, all the same, all right? Depolarization is going from -90 towards zero, all right? That's depolarization. Repolarization is returning back to -90, all right? If you think about decaffeination, you're removing from whatever it was, all right? So depolarization is going less negative, all right, becoming more positive. Repolarization is returning to -90 or returning to your resting potential. And with these, I guess you also should know, the depolarization sodium channels are open. Repolarization, potassium is leaving the cell. Excitation, contraction, coupling, you guys, all right? These are the events that transmit the action potential along the sarcolemma and are coupled to the sliding of the myofilament. So here's what excitation, contraction, coupling is. Excitation is what happens between the nerve and the motor end plate. All right, that's excitation. Excitation, contraction, coupling is when it moves from the motor end plate to the T-tubule. So as it's moving along the sarcolemma, that's excitation, contraction, coupling, and that's what we want to try to do in 10 minutes. The action potential is propagated along the sarcolemma down into the T-tubules where voltage-sensitive proteins and tubules stimulate calcium release in the sarcolemma. Particular calcium release leads to contraction. Action potential is brief and ends before contraction is seen. So to answer your question, you were saying, is that action potential that we saw, that peak, was that the actual muscle contraction? Not exactly. It's brief. So the action potentials are occurring before we see the contraction in those milliseconds. So here is excitation, showing excitation, contraction, coupling. Here's the nerve. The signal that's moving along here, this little red line, that's coupling or connecting excitation to the contraction. This is showing you again a T-tubule, terminal cisternae on either side. Here's, I think, a better picture. So, hits the nerve. Nerve has the contraction in the neuromuscular junction. It spreads along the sarcolemma. Here's down the T-tubule. On either side of the T-tubule is the terminal cisternae. Causes calcium to be released. The calcium that's released attaches to troponin. You can see your little blue thin filament here. So we're kind of tying everything together. And then that moves troponin where myosin can attach to actin. And it creates the cross bridge, and the cross bridge allows for the shortening of the sarcolemma. And this is the steps there. Oops. All right. You can't read that, it's so small. So the video, ready for the video to show the coupling of excitation and contraction? And then I do my short answer question. That might have took less than 10 minutes.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Last thing I want to show.
Membranous network.
Now, the T-tubules, here's what I want you guys to kind of see. When you're looking at the diagram here, the T-tubules are wrapped around every myofiber. So remember we saw a muscle cell, thousands of myofibers inside a muscle cell. One T-tubule, it branches and spreads and loops around every myofiber. So when that signal goes down the T-tubule, it comes in contact with all the myofibrils at this specific point. And the signal keeps spreading to the next T-tubule, contracts there, and keeps spreading. So that's what I kind of want you to understand, is that signal is going down the T-tubules, it's coming in contact with each myofiber. All right. And then of course that causes the release of calcium. Hopefully you guys are kind of was coming back to the beginning and making connection, because you saw the triad here. Remember you saw the triad in the beginning of the chapter today? And this is what you guys have to know. You ready for your question? You guys just like, let me go, please. Free me,
I just want
right?
to see later.
Huh?
I just want to see how you explain this.
How I explain it?
Yeah.
Okay. All right. So your short answer question will say something like this. Please define or explain skeletal muscle contraction using the following terms. Your answer should be something like this. The brain sends a signal down a motor neuron. When the signal reaches the end of the motor neuron, calcium comes in to the axon terminal and causes the release of acetylcholine. Acetylcholine diffuses or floats across the synaptic cleft. When acetylcholine attaches to the ligand, L-I-G-A-N-D, ligand-gated receptor, the ligand-gated receptors open. Sodium goes in and some potassium comes out. A whole lot more sodium goes in than potassium goes out. This begins to change the membrane potential of the sarcolemma and the muscle cell. This is the beginning of an action potential. The action potential now spreads from the neuromuscular junction around the sarcolemma. When it reaches a T-tubule, it goes down the T-tubule, comes in contact with the terminal cisternae, which are an extension of the sarcolemma, which are an extension of the sarcoplasmic reticulum, and the sarcoplasmic reticulum then release calcium. The calcium attaches to troponin, removes tropomyosin, and allows for the myosin heavy chain to attach to the actin thin chain, which causes the shortening of the sarcomere. The end.